This is Chapter 4, Part 1, The Functional Anatomy of Prokaryotic Cells. So here's our learning objective for this. Make sure you take a moment to take a look at this. So the average size ranges from about 0.2 to 2 micrometers in diameter and between 2 and 8 micrometers in length. Most bacteria are monomorphic, meaning they can only have a single shape, but a few are pleomorphic, which means they can take on many shapes. So we also have several different general types of shapes that we see. We see bacillus, which is rod-shaped. We see coccus, which are going to be spherical. Spiral, we can see Vibrio, Spirillum, and spirochetes. We can have star-shaped, and we can have rectangular bacteria. So here you can see some Vibrio. Vibrio are going to have a, kind of a shape that can take on any kind of shape, essentially. Generally, you see them almost as kind of like curved rods, but for the most part, they can be almost any type of shape. Spirillum, they're going to be in spirals, and spirochetes are going to be long, skinny spirals. The star-shaped bacteria, you can see them here. So they do have this kind of star appearance, and so these are very easy to recognize when you see them. Then you have rectangular bacteria, and so these are very interesting as well. Anything that has a kind of regular shape is almost really cool in biology. So we can also have pairs where we have diplococci or diplobacilli. Clusters will be staphylococci. Chains will be streptococci or streptobacilli. Groups of 4 can be tetrads. Cube-like groups of 8 are considered sarcanae. So you can see all of these here. So you can see the diplococci and the streptococci, and then you can see how they start to go into clusters, which is going to be the streptococci, and then you can see the sarcanae as well. So the single bacillus, so this is going to be a rod-shaped bacteria. The coccobacillus is going to have a kind of rounded off rod shape. The diplobacilli, these are going to be when there's 2 of them, and the streptobacilli are going to be long strings of these. So the scientific name is bacillus, and the shape is bacillus. So this is how we kind of name things a lot of times when we see stuff in the microscope. This is how we come up with names. So here you can see some bacillus, and so you can see very long lines of these. So this is probably some type of streptobacillus, or so Bacillus anthracis is this particular one. You can also see where it's also starting to form spores, as Bacillus anthracis is one of those that does form spores. So here's your check your understanding question. Make sure you take a moment to take a look at this. So the structure of a prokaryotic cell. So you can see in this image, and you can see the very long flagella, and so a lot of the other parts of this image are very, very small. So I'm going to try to go through them piece by piece with you over the next several slides. So here are the learning objectives for this next step. So let's make sure we take a moment to take a look at these. So the glycocalyx is external to the cell wall. It's very viscous and gelatinous. It's made of polysaccharides and or polypeptides, and we've got 2 types. We've got the capsule, which is neatly organized and firmly attached, and then we've got a slime layer, which is going to be unorganized and loose. And so the glycocalyx contributes to the virulence of a pathogen. The capsules prevent phagocytosis, and extracellular polymeric substances can help form biofilms. And so here you can see the capsules of Streptococcus pneumoniae. This is the cause of pneumococcal pneumonia, which is a very, very rough pneumonia from what I understand. And so you can see the capsules on these. You can see kind of how they glow around the bacteria, and this is going to help it to form biofilms as well. The flagella, the filamentous appendages of the external cell, these propel the bacteria. They're made of the protein flagellin. And we've got 3 parts. So we've got the filament, which is the outermost region. We've got the hook, which attaches to the filament. And we've got the basal body, which consists of rod and pairs of rings. This anchors the flagellum to the cell wall and the membrane. So here, the structure of a prokaryotic flagellum. This is when you've got the cell wall as well as the plasma membrane, and the parts and attachment of the flagellum of a gram-negative bacterium. So it's going to be a little bit different in gram-positive. In gram-positive bacterium, you only have the single cell wall and the plasma membrane. And so the parts of attachment of flagellum in a gram-positive are a little bit different. And so here you can see the different arrangements of bacterial flagella. You can see the peritrichous, which is all around. You can see the monotrichous and the polar. And then you can see the lophotrichous and the polar. And then you can see the amphitrichous and polar there. So flagella allow bacteria to move towards and away from stimuli. This is what we call taxis. And so flagella rotate to run or tumble. And so flagella proteins are H antigens and distinguished among serovars. So like Escherichia coli, O157H7, we know this one is a deadly one, based on, this is the one that got a bunch of people sick probably about 2 decades ago. But this is how we distinguish among them. So bacterium running and tumbling, notice the direction of the flagellar rotation. The blue arrows is this. And so you can see the flagella all around it. And so these are going to kind of run, they're going to kind of spin around, and then they're going to kind of start to unwind so that they can tumble, and then they're going to run again, and they're going to tumble, and they're going to continue to tumble. And so the gray arrows indicate the direction of the movement of the microbe. And so you can see this as you continue to go around the circle. So this is a Proteus cell. In the swarming stage, may have more than 1,000 peritrichous flagella. That's a lot of flagella. And so normally we only see cells with only a few, but generally when you have a lot of flagella, it's very hard to kind of coordinate that motion. So axial filaments are also called endoflagella. These are found in spirochetes, and they're anchored at one end of the cell. And this rotation causes it to move like a corkscrew. So here you can see the axial filament, and so this is a photomicrograph of the spirochete Leptospira showing an axial filament, and it's going to help it move kind of like a corkscrew. And so here you can see a kind of a cartoon diagram of this and how it's kind of wrapped around there. So fimbriae, these are hair-like appendages that are going to allow for attachment. If you were a bacteria, you would want to have some way to attach yourself to things so you didn't fall off of something and end up in a place where you weren't going to be able to survive. Pili, these are involved in motilities, both gliding and twitching motility, and so the conjugation pili are involved in DNA transfer from one cell to another. So here's your check your understanding questions for section 3. Make sure you guys take a moment to take a look at these. So next we're going to cover the cell wall, and so we're gonna look at these different objectives, and we're going to go through all of this. So the cell wall permits osmotic lysis and protects the cell membrane. It's made of peptidoglycan and bacteria, and it contributes to pathogenicity. So here you can see the capsule and the cell wall, as well as you can see a lot of other things in this transmission electron micrograph. And so this is going to be one of those interesting things that we see, where these bacteria actually have a lot of stuff going on inside of them. And so the peptidoglycan is a polymer of a repeating disaccharide in rows. So you have N-acetylglucosamine, or NAG, and you have N-acetylmuramic acid, which is NAM. And so rows are linked by polypeptides. And so here you can see how they compare and contrast to each other, and they are linked, they are joined as in a peptidoglycan. So these bacteria cell walls, like I said, they are joined. They alternate, and then they are joined together, and this helps them to make a very structured cell wall. So gram-positive cell walls are very thick. They're made of teichoic acids. What you're going to see in gram-negative is going to be a very thin peptidoglycan, an outer membrane, and a periplasmic space. The teichoic acids, these are lipoteichoic acid, links the cell wall to the plasma membrane, and the wall teichoic acid links the peptidoglycan. These are going to carry a negative charge and regulate the movement of cations. Polysaccharides and teichoic acids provide antigenic specificity. And so here you can see a wall with these teichoic acids, and you can see all of these different things in here, and you can see the plasma membrane down there at the bottom as well. So gram-negative cell walls are going to have a periplasm between the outer membrane, and their plasma membrane contains peptidoglycan. The outer membrane are made of polysaccharides, lipoproteins, and phospholipids. So gram-negative cell walls, these protect from phagocytes, complement, and antibiotics. They're made of lipopolysaccharides, so the O polysaccharide functions as an antigen, like in the E. coli O57H7, and the lipid A is an endotoxin embedded into the top layer. The porins, or the proteins, form channels through the membrane. So one thing I'll mention about the lipid A, if you ever try to kill gram-negative bacteria that's in a systemic infection, you have to be very careful how that you use antibiotics for this, because there are gram-negative specific antibiotics. However, when you when you lyse these cells, it releases this lipid A, which is an endotoxin, and it will send the patient into shock. And so the bacteria cell walls, you can see this is the gram-negative cell wall. So you have essentially two membranes separated by that inner membrane with the peptidoglycan. So cell walls and the gram staining mechanism. Crystal violet iodine crystals form inside the cell. The gram-positive, the alcohol dehydrates the peptidoglycan, and the crystal violet 1 crystals do not leave. So the gram-negative bacteria, the alcohol dissolves the outer membrane and leaves holes in the peptidoglycan where the crystal violet washes out, the cells are colorless, and then the safranin adds the stain to the cells. And so here you can see the difference between gram-negative and gram-positive. Generally, gram-negative, you're going to see more cocci, and gram-positive, you're going to see more rods. But that's only a kind of an in general kind of thing. Don't take that to be a rule of thumb. So in gram-positive cell walls, there's two ring and a basal body of flagella. They produce those endotoxins, and they're highly susceptible to penicillin. These can also be disrupted by lysozyme. In gram-negative cell walls, we've got 4 rings in the basal body of a flagella, and they produce endotoxins and exotoxins. They have a very low susceptibility to penicillin. So acid-fast cell walls are like gram-positive cell walls. They have a very waxy lipid or mycolic acid bound to the peptidoglycan. These are what we find in the Mycobacterium and the nocardia. These generally stain with carbonyl fusion. So here you can see this is Mycobacterium tuberculosis, and these are going to stain with that carbonyl fusion. The mycoplasms lack cell walls, and the sterols in the plasma membrane and the archaea are generally wallless, and so they have walls of what we call pseudomurin, or they lack the NAM and the D amino acids. The lysozyme hydrolyzes bonds in the peptidoglycan. The penicillin inhibits those peptide bridges in the peptidoglycan. A protoplast is a wallless gram-positive cell. A spheroplast is a wallless gram-negative cell. And protoplasts and spheroplasts are susceptible to both to osmotic lysis. The L forms are wallless cells that swell into irregular shapes. So here's your check your understanding questions. Make sure you guys understand what's going on here. And if you don't, ask me a question. So the structure's internal to the cell wall. So the phospholipid bilayer that encloses the cytoplasm is going to be that plasma membrane. And so the peripheral proteins on the membrane surface are going to be part of the plasma membrane, and the integral and transmembrane proteins penetrate this membrane. So here we can see the plasma membrane, we can see a micrograph as well as the cartoon image of it. So here you can see the peripheral and the integral proteins that are either going to cross the membrane or they're not going to cross the membrane. And so these are proteins that help the cell do work. So this structure is really a fluid mosaic model. A lot of people think that this is a static structure, the cell membrane. It's always moving, it's always changing, and so they can move freely in various functions, for those various functions. And so the phospholipids rotate and move around laterally, and it's also self-sealing, so if there is a hole in it, it will try to seal it up as fast as possible. The plasma membrane has a selective permeability that allows the passage of some molecules but not others. It contains enzymes for ATP production, and some membranes have photosynthetic pigments on the foldings called chromatophores. So here you can see those chromatophores, and so they can turn different colors. The damage to the membrane by alcohols, quaternary ammonium, like detergents, and polymyxin antibiotics causes leakage of cell contents. Passive processes in the cell membrane are when substances move from high concentration to low concentration and no energy is expended. Active processes are when substances move from low concentration to a high concentration and energy has to be used in order for this to happen. So simple diffusion is a passive process, and it's the movement of solute from a high concentration to an area of low concentration. And it continues until the molecules reach equilibrium. So here you can see an example of that passive process. And so facilitated diffusion is also a passive process. A solute combines with a transporter protein in the membrane, and the transporter transports ions and larger molecules across the membrane with the concentration gradient. And so here you have these very specific transport proteins, and so this is going to be an example of facilitated diffusion. Osmosis is the movement of water across a selectively permeable membrane from an area of high water to an area of lower water concentration. Generally, this is through a lipid layer, and the water channels that we use for this are what we call aquaporins. So osmosis through the lipid bilayer and an aquaporin are both here. Osmotic pressure is the pressure needed to stop the movement of water across a membrane. And so here you can see this, and so you might have done this in lab, depending on where you took biology. But at the beginning of an osmotic pressure experiment, the bag in there, that selectively permeable membrane, is not full of water yet. And so as you continue to have that go down, you'll see that glass tube fill up with water and solutes because the water is going to start to push into it because of the more solutes that are in there. So an isotonic solution is a solute concentration equal on the inside and the outside of the cell. So the water is at equilibrium. The hypotonic solution is when the solute concentration is lower on the outside of the cell than the inside of the cell, and so water is going to move into the cell. And a hypertonic solution is going to be the solute concentration is higher on the outside of the cell than the inside of the cell, and the water moves out of the cell. And so this is an issue when you have a cell wall. Okay, so you guys might remember this from anatomy and physiology or from basic biology, where the isotonic solution, everything's fine. The hypotonic solution, the cells are eventually going to swell and they're going to go through osmotic lysis. They're going to rupture. But the hypertonic solution is when the water moves out of the cell, causing its cytoplasm to shrink, and this is what we call plasmolysis. Active transport is an active process, and it requires the transporter protein and ATP, and generally this goes against the concentration gradient. Group translocation requires the transporter protein and the phosphoenolpyruvic acid, PEP, substance is altered as it crosses the membrane. So here's your check your understanding questions. The cytoplasm is the substance inside the plasma membrane, and 80% of water plus proteins, carbohydrates, lipids, and ions, as well as the cytoskeleton. The nucleoid is the bacterial chromosome. This is a circular thread of DNA that contains the cell's genetic information. And the plasmids are extrachromosomal genetic elements that carry non-crucial genes like antibiotic resistance and production of toxins. And so these plasmids can be destroyed and nothing really happens to the cell. So the ribosomes, these are the site of protein synthesis. They're made of protein and ribosomal RNA. And so we talked about the ribosomes in the eukaryotic section. And so the 70S ribosomes in bacteria are made up of a 50S and 30S subunit. So you get these two together, and the complete ribosome is a 70S subunit. We also have inclusions. We have metachromatic granules like volutin. These are phosphate reserves. Polysaccharide granules, these are going to be energy reserves. Lipid inclusions are going to be for energy, as well as sulfur granules for energy. The carboxysomes, these are going to have Ribisco enzymes for CO2 fixing during photosynthesis. The gas vacuoles are protein-covered cylinders that are going to maintain buoyancy, and magnetosomes are iron oxide inclusions that help to destroy hydrogen peroxide. So here you can see some magnetosomes, which are really cool because they hold onto that iron and they can react to magnetic fields. The endospores, these are resting cells produced when nutrients are depleted, and so they're resistant to desiccation, heat, chemicals, and radiation, and they're generally produced by Bacillus and Clostridium. The sporulation is the endospore formation, and the germination is the endospore returns to a vegetative state. So here, the endospore of Bacillus subtilis, and so the whole process here is outlined here. So the spore begins, the spore septum begins to isolate the newly replicated DNA and a small portion of the cytoplasm. The plasma membrane starts to surround the DNA cytoplasm, and the membrane is isolated in step 1. The spore septum surrounds the isolated portion, forming the forespore, and so then you have 2 membranes around that that DNA there, and the peptidoglycan layer forms between the membranes, and then the spore coat forms, and that spore coat is very tough, and then that endospore is freed from the cell. So here's your check your understanding questions. Please make sure you take a moment to take a look at these.